I have a few announcements to do for today. The first one is who here is not going to be able to do the exam on the 23rd, except from those that told me already. Anybody else celebrating Passover? Okay, so for the people that didn't show up today and watching this on YouTube, please send me an email before the 31st of March so that I can include you in the alternate exam date, okay? Everyone else, it's going to be on the 23rd as scheduled. For the others, it will be on the 19th, so the Friday before Passover week, okay? If you have anything on that day, I can't accommodate twice in a row. You're gonna have to figure out something with that, okay? So that's for that. Next week's tutorial on Monday will not happen because we have a long weekend, right? The Easter weekend. So if you have a tutorial on Monday and you still wish to attend the tutorial, then I suggest you go to the Wednesday section, okay, of your TA. So if you're from 1:30 to 3:30 on Monday, try to go on Wednesday. The quiz will be on Moodle online, so everyone is gonna get it at the same time, either on Wednesday or Thursday, okay? Any questions up to now? Yeah. I will, I think it's on the course outline, but just to make sure, I will send an email with everything, okay? Yeah. Online, exactly. Everyone's gonna do it online. I will send an email about that too, okay? Yeah, so that's that. What else did I want to tell you? There's 2 weeks left of classes, or 2.5, let's say. Last day of class for us, I think it's on the 14th. No, it's on the 8th, 9th, 11th, exactly. Somewhere somewhere along those lines. I think the 14th is the last tutorial, probably, or something like this, right? The 11th. So 15th is a Monday? Okay. So then that day, I guess you have nothing? Nothing? Huh? Let's see. So next tutorial, you'll have a quiz, and then the week after you have a quiz, and that's it. Okay. So no tutorials on the week of the 15th, okay? Let's do it this way. So you have a tutorial next week. Everyone has, well, not has, but if you want to, you go on Wednesday, because Monday we don't have class. The week after, back to normal tutorials in the tutorial room with the quizzes in person, okay? Make sense? Any questions for me? There's question number 1 of the midterm exam. Has 2 potential answers, so I'm going to go over all the exams again and give points to those who answered the other answer. So it seems like free points everywhere, right? It's okay. When you make the exam, sometimes you don't think about you don't think about it in a specific way, and then you end up finding that it works too. Okay, so I'll be doing that update for the midterm. So you should be getting a new Excel sheet with the grades. Take a look at it. If your grade changed, well, good job. If not, then can't help you, right? Okay, so the serious stuff. So today we're going to finish the regulation of gene expression. Thursday we'll start recombinant DNA. That means next week's next week's quiz will be on protein synthesis and the first part of gene regulation, okay? The quiz in the week after will be on the first part of recombinant DNA, as well as these slides here that you're going to see today. Yeah. Yeah, that will be the week. That should be this week. When is your tutorial? So what did you do yesterday? Oh, protein, you mean? Yeah, so that will be next week where you'll cover this, because remember we do like one lecture and then practice in the tutorial, and then the week after is the quiz on that. Yeah, just 1 sec. Yeah, you're good? Kind of. Man, this is really mixed up. Okay, let me just check this. Okay, it's getting out of hand here. I will take a look at it. Okay. Yes, Isabel.
Yeah, I was just wondering, so then, is it whatever we did in the last tutorial is on the next week's tutorial?
What?
I mean, sorry, I mean on the next week's quiz?
Exact.
Okay.
Technique. Yeah. So if you've practiced something, just next week is the quiz on it. Okay. Just do that. Anyways, I'll verify what you're saying there, but I'm pretty sure I did it properly this time. We'll see. Okay, so regulation of gene expression. So we looked last week at the lac operon. Hopefully you had time to take a look at that and understand it a little bit more in depth, because today we're going to add a few layers of complications or complexity. So I just want to go over the lac operon. I'll use this slide here, but you know, it's the same thing. So the lac operon has a promoter, operator, the LAC-Z gene, the LAC-Y, and LAC-A. In the context of our course, we don't really care about the A gene, just that it encodes a transacetylase. The rest doesn't matter, and it's an enzyme that modifies one sugar into another through a transacetylation step. Doesn't really matter all that, just know that LAC-A, transacetylase, okay? LAC-Z is beta-galactosidase, LAC-Y is permease. The role of permease is to allow lactose to enter the cells, while beta-galactosidase is the enzyme that will break a lactose in galactose and glucose. Cells love glucose, so they will try to do anything to get to glucose, and this is one way where this operon actually acts as a sensor for lactose to then break it down to glucose, which is then used into glycolysis, okay? So if you remember properly, there's another gene called the I gene. It's the inhibitor gene. So this protein, when it's made, it will bind to the operator sequence and prevent transcription of the operon, okay? Because RNA polymerase that binds to the promoter cannot move because there's something blocking it here. Obviously, there are variations of this. We saw the OC operator, which is the constitutively active operator. This operator cannot be bound by either I plus or the super strong version of I, which is the IS, okay? Because both don't recognize the sequence of the OC operator. We also saw the O minus, which is pretty much like the OC. The only difference is when we're talking about the gene itself. O minus, you don't have the sequence, while OC, you have alterations in the sequence that prevent it from being bound by the inhibitor. Obviously, if it's P minus, that means you don't even have the promoter, so no RNA polymerase can bind, or you have mutations in the promoter, for example, in the TATA box that prevents binding of the promoter. If you have Z minus, it means that you either encode a protein that is non-functional, or you just don't have the Z gene, and so you won't make the Z gene, okay? So when you're asked if you will be making the beta-galactosidase based on a certain, based on the presence of lactose and whatever configuration you have, if you have Z minus, doesn't matter if you're inducing not OC, not whatever, okay? You're not making the Z. And we also looked at another thing, is when you have two diploids, when you have a diploid. So when you have mating between two haploid bacteria, each one will bring its own operon, and so you have to think about cis versus trans-acting things. For example, the I protein is trans-acting, because if you have a diploid with two different operons, and let's say one of them has I minus, but the other one has I plus, that I plus, because both operators are in the same cell, that I plus can act on both operators, okay? And this is what I mean by trans-acting. So we're talking about trans-acting proteins, which is the I protein, and we're talking about cis-acting sequences, which is the operator sequence. Yeah.
So like the RL makes the repressor, and then it's like the repressor is the protein that can bind on the protein?
Yeah, because it can travel in the cell and go and bind on the DNA, both DNA, okay? Usually what happens when you're getting a question about a diploid, you'll have a situation where one of them will be O minus, for example, or OC, and the other one will be O plus. And you'll have, for example, the one that's OC, you'll have Y plus, but Z minus, and then you have to figure out, based on those configurations and the presence of lactose or not, what is transcribed and what is not, okay? I think I gave an example last lecture. I guess I can give another one really quickly, and then we can get into the next step. Okay. Let me just see this camera here because, okay, so basically I can't from here. Okay, just put this here. Because when I recorded last lecture, some of the text doesn't appear, okay? So let's see. Okay, so when you have a diploid, usually you'll end up with a situation where you have I minus, and then I something, either plus or S, doesn't matter. And you'll end up with P, O, Z, Y. And let's even say A here if we want to, but it, same principle, just, you know, doesn't really matter, okay? So then P, O, Z, Y, A. And let's say, for example, this one is OC, like this, and then Z will be plus, and this will be minus, minus, Z will be minus, this will be plus, plus. Okay? So this is what a exam question looks like, okay? So what you'll have is a situation like this, and then you'll be asked, based on induction or not, what protein of these three will be made. So in general, when you have an OC here, that means induced or not induced, it doesn't matter. You will be making Z all the time, okay? Beta-galactosidase. This one here does not have any effect, so we forget about it. This I plus is trans-acting, so it can come here, but because it's OC, that doesn't work. Or it can come here, and because this is O plus, then it's actually inhibiting. So then you have a situation where if it's not induced by lactose, you won't be making Y or A, because this one here can't make them, and this here requires induction. And when you have lactose, you will be making Y and A plus. Okay? Good. So now the level of complexity that I'm going to add here is the positive regulation of the lac operon. Okay? So the lac, usually bacteria, actually pretty much anything, they prefer glucose to anything else because it's simpler and it has a full dedicated pathway to breaking down glucose and getting energy from it. Okay? So when cells have glucose, they act differently than when they have lactose. Okay? One thing that you have to remember is that the end goal of all of this is to get energy, right? So when you have low levels of glucose, you will be breaking down the amounts of ATP, which you probably learned in CJAP, as your energy, into cyclic AMP. First it will be ADP, AMP, and then CAMP, okay? If you have high levels of glucose, that means your body already has too much energy and it will not need to convert ATP to CAMP. So it will kind of decrease this sort of conversion here. Okay, so this is the RL for inhibition, but it's not really inhibition. It's just decrease the amount of ATP that is converted to CAMP. Okay, see it like this. This is going to be simple for you to remember. Not enough energy, you use the ATP. Not enough glucose, you use the ATP that you have. High glucose, you do use it, but just imagine it as you're not. Okay, it's going to be simpler like this. So in this case, when the cell has, so let's start by this here. So there's this protein here called the CAP, catabolite activated protein. for you to remember it, you can also remember it as the CAMP activated protein, because this protein here requires CAMP to be in its active form. Okay, it's a dimer. That means it has two subunits, and each subunit has a spot for CAMP molecules. So in a way, it acts as a sensor for CAMP. So when there's CAMP molecules floating in the cell, that means there's low glucose, and so because there's low glucose, the cell will look for other sources of energy, right? And so when there's low glucose, the CAMP will activate the lac operon because it will kind of make it turn lactose, another source, into energy, okay? And so what happens is that this CAMP cap protein, so this complex protein here, can bind to the promoter. And by binding to the promoter, it will enhance transcription of the lac operon. The opposite of this is when there's no CAMP. In that case, these cap protein cannot bind to the promoter because they are in a conformation that does not allow them to bind the promoter. Okay, we'll see what this means in a second. So in the promoter, we learned that there is a region for the RNA polymerase, which usually will look for these sequences, that -10 and -35 sequence, but there's also other sequences that we didn't talk about. One of them is the cap binding site, and this here is found in the promoter of the lac operon. So this is where the cap will bind, and you'll have RNA polymerase that binds here, and they will interact with each other. By interacting with each other, they will affect the ability of RNA polymerase to transcribe faster or at a lower pace, and I'll show you that in a second. So the way this is done, if you take a look at the ball model of this protein, you have the two subunits that are here, right? And then you have the space for CAMP binding. When CAMP binds, it will change the conformation of the cap protein, and then that will bend the DNA in a way, okay? By bending the DNA, it will kind of bring RNA polymerase closer to it and improve and interact with it, which will improve its ability to transcribe the lac operon. Okay. Do you have any questions? Easy? Good. So when there is glucose, we don't really care about lactose, because glucose is much better than lactose. It does not require a breakdown into galactose and glucose to get the energy. So it's more efficient to use the glucose than to activate pathways that will break down other sugars. I think this is simple, very intuitive. And so when you have glucose present, there's no CAMP. And because there's no CAMP, this cap protein will not be active and will not be able to bind the promoter. Okay? At the same time, if there's no lactose, the I protein will also bind the operator, and you'll have kind of a double inhibition of the lactose, the lac operon, I mean. Okay? Works up to now? Yes. Okay. So that's that. So in the case where there's still low CAMP, but you have lactose, then you can induce the lac operon like we saw last lecture. And this will give you a small amount of lac mRNA and will produce a small amount of lac Z, lac Y, and lac A. If you don't have glucose and you have lactose, then you need to focus. Yeah.
Sorry, there's very little lac because
Yeah. So in general, the lac mRNA here, I'm comparing it between two situations where you have glucose and lactose, and when you have just lactose, okay? And so there's a different need for the lac operon. If you have glucose and lactose at the same time, you will prefer glucose, right? So you'll have very little lac mRNA being made. If you have no glucose and lactose, then your only choice is to use lactose, and in that case, you'll need a lot more lac mRNA to take care of it, okay? In the situation where you have a lot of glucose and lactose, then you can imagine in your head, just in the whole cell, that all the proteins for glycolysis will be like super active, right? Okay. So back to this. So in the case where you have no glucose, that means your CAMP is high, and some lactose. So in between both cases, lactose is present. It's just the level of glucose that is different. Then because you have CAMP, it can bind the cap protein, which you can remember again as the catabolite activated protein or the CAMP activated protein. CAMP will bind to it, change its structure, and then that structure will be able to bind the promoter. And then because there's lactose, you'll be able to remove the inducer, the repressor, I mean, from the operator, and this will make a lot more lac mRNA. Is that easy? Not too bad? Okay. Do we do a question like this? Yeah, you know what? Why not? Okay, let's do a question on the board. So a question for this type of problem can look basically like this. So you have you know what? Let's mix you up there. Okay, so P plus, O plus this, and these are all plus. So first we have this, and then I tell you that I add, let's see, low glucose. So there's low glucose. Low glucose and high lactose. Okay, so if you have these parameters here, can you tell me if you're going to make Z, Y, and A? Are you? IS will bind here, so it doesn't matter what you have there, okay? Because you have IS binding here, doesn't matter if you have low glucose, high glucose, in between, lactose, doesn't care, okay? If you have this situation here now, if you have a situation like this, are you going to make this? Sure? So because you have low glucose, does that mean you have high CAMP? Okay. More CAMP, right? Yeah. So CAMP protein there, just make square like this will bind here. So your CAMP activated protein will bind here. You have high lactose, so this will be removed from here. This will be, oh, there it is. So that will be removed from there, and so you'll be making a lot of the sequence here, ZYA, okay? And then you can imagine a lot of different combinations of this. Yeah?
If it was I minus, is it the same as IS?
No, I minus means that you don't make any protein. So there's no I, and so this is always free in the haploid state. In the diploid state, you have to look at the other I protein from the other cell. Cool? Okay. Yep. Exactly. So if you have I minus here, the glucose level is what matters. The lactose doesn't matter anymore because you don't need to induce, to repress the repressor. Yeah?
And I think last class you said something like P minus. Does that mean like nothing happened?
Exactly. When you have a problem in the promoter, so I'll show you that here. Now, obviously, this is like a top view, but in reality, if you have a problem in the promoter, some mutations will be here. So that means the RNA polymerase cannot bind. Some mutations will be in the cap site, binding site, so that means the cap binding site will not be bound. And so we just assume that at this level that P minus means all this region here does not exist. And so RNA polymerase and cap cannot bind. Cool? Simple? Good. Okay. So, a small little review of the past two, the beginning of this lecture and the past one, and then we'll go to chapter 12, which we have some information about more regulation of gene expression, and that would be it. So, there are two mechanisms, as I said last lecture, repression and activation. In repression, you have a repressor protein which will bind to a certain operator and prevent transcription. You also have an inducer which will bind to the repressor and repress the repressor, okay? In other words, it will cancel the effect of the repressor, and so it will allow the transcription of the genes in the operon, okay? In our case, we learned about LAC-I, which is the repressor, and the inducer is lactose, okay? Now, for the activation mechanism, it's kind of the opposite. When you have the activator, it needs to be induced by the inducer to bind a certain sequence which will allow for transcription of those genes, okay? And then if you don't have the inducer, then your activator is in an inactive form, and so it cannot promote transcription, right? In the book, if you continue reading, you'll get into the arabinose operon and other operons down there. If you want to read them, that's okay. It's going to give you a better image in your head about this stuff, but they're not necessary, okay? It's just more for you to understand the whole thing. Yes, Nicholas? That's it. Exact. Good. Yep. No. Yep. Yep. It's just random letters they use to. No, you have the promoter. I don't know what the I is here, but I guess the induction site or something like this. Okay. Okay, transcription factors. This is basically my whole research right now. Okay. So, transcription factors are an additional way to regulate transcription of genes. So, we've studied that there are many levels of gene regulation. We looked at DNA, right? And we'll look a little bit more into the DNA because we'll be looking at nucleosomes and histones and all that in a second. We also looked at the transcription, which has a higher level of regulation. For example, alternative splicing. So you can have different forms coming from the same mRNA based on which introns and which exons you keep and which you don't. So that's alternative splicing. We also have the cap, 5', the poly-A tail, etc. Okay. Translation, we also have different levels of regulation of the expression of genes. For example, phosphorylation, ubiquitination, which promotes the degradation of the protein, etc. Okay. And then there are mutations that make the protein more performant, less performant, or not performant at all. Okay. So it's not just like Mendel said it, you have the gene, then the allele, then you're expressing it, and all that. Actually, most of the genes that you have in your body are not active, and they get activated only in certain situations. I guess if they were all active, you'd be looking kind of weird. It would be a little bit messed up, okay? So Mendel was kind of right, but not 100%. Now we know a lot more about this. And now I'm going to add another level of regulation, and this is at the level of transcription factors. So in your body, you have, so in your cells, you have 20,000 genes, okay? And these 20,000 genes are actually active in certain cells, and while groups of these 20,000 genes are activated differentially between cells, okay? You don't have the same genes that are active in your liver cells compared to your neurons, okay? And this is mostly because of these transcription factors. So these transcription factors are proteins that are expressed in specific cells, not all the cells, but specific cells, and the different ones will kind of promote the transcription of a whole program related just for those cells. Okay. For example, your liver has its own transcription factor, and its liver. Let's go with the, I forgot the 3-letter code there, but let's go with, for example, the ovaries, okay? So the ovaries have their own specific transcription factors, and those level of transcription factors will be the ones that determine the genes specific for the ovaries, which is, for example, the estrogen receptor, and there are other receptors, okay? In my research, I'm studying the androgen receptor, the estrogen-related receptor, which is kind of the estrogen receptor, and many others which I won't get into, but there are hundreds of them, okay? And so if you take mechanisms of development, you'll learn a lot about this, and you'll see how you start from one cell, and then because of the gradient of transcription factors along the cell, then they will divide into two, but this one has more of one of the transcription factors, and it will lead to making a specialized cell later on that is related, for example, to your legs, right? While the other one, because it's on the other side, does not have that much transcription factor, and so it will become your head, right? And then they get developed based on these gradients, okay? So what concerns us is really where they bind and their effect on transcription. And so promoter, core promoter means the same thing, okay? So you have the core promoter where you have just proteins that bind there, and these are called general transcription factors. These are pretty much found in every promoter, and their role is just to bring things together. That's it, okay? Then you have also two other types of regions on the DNA. You have the proximal enhancer and the distal enhancer. Proximal enhancer is right next to the core promoter, okay? And there you'll have specialized transcription factors that will be binding. And the distal, same thing, just further away from it. Usually the distal is around 20,000 base pairs away from the promoter. So you can see how far it can be. And this is important because it will play with the structure of the DNA and kind of turn it into a loop here, which will allow these transcription factors to bind to these ones and then bring RNA polymerase. We'll see this in a second. I'm just giving you a quick overview. Okay. So in my case, for example, as I'm studying the ER alpha protein, which is the estrogen-related receptor alpha, it binds to an enhancer of most of your energy regulation genes. It's also very active in cancer, and so I'm trying to mutate it to see if it binds here in a stronger way, and if those mutations will activate transcription more or less, because these mutations were found in patients that have triple-negative breast cancer. So I'm trying to see the effect of the mutation on the transcription of these genes, okay? And a lot of research is done on these transcription factors. It's a huge field. So maybe one day one of you guys will be working on this. I don't know. But yeah, so that's that. So now what's the role of these enhancers? The enhancers, their role is just to kind of bring the whole transcription machinery together so that the transcription can happen. For example, for the distal enhancers, they will be usually bound by a transcription factor and many other proteins, including the mediator protein and the transcription factor 2D protein. And these here serve just as a scaffold kind of to bring this to this. And so it will create like a bridge between the enhancer and the core promoter, and that will allow for the proper placement of RNA polymerase 2 on the core promoter and transcription to happen, okay? Now the enhancer is just not one protein bound there. There's usually like tens of proteins that are bound there, and there are two different types of proteins that can bind there, either co-activators or co-repressors. So usually you would have a transcription factor or two, with their own co-repressors or co-activators. If you had the co-activators, this structure here will be even better, and you'll have more transcription. But if you have co-repressors, it will disrupt this structure here, and you'll have less transcription. So this is how enhancers kind of control transcription of genes. Cool. What else? I think I said that. So the transcription factors, they also recognize consensus sequences, okay? And usually they're called, no, not usually. They are called palindromic sequences, meaning that you can read it. So it has basically the same word read from both sides, okay? So for example, let's see. So you have CACAC, CACAC. Another palindrome that you all know is Laval. You can read it from both sides, so you can remember what a palindromic sequence means by remembering Laval, okay? And so what they saw, scientists, is that when they mutated these palindromes, they found that there's a decrease in transcription levels. And so this is just to show how important these enhancers are. If you remove this sequence, your enhancer cannot be bound, and because it cannot be bound, you lose the transcription. So this is another layer of regulation. Yes, Isabel.
Sorry, where is the column from?
CACAC, right? ATATA. Make sense? Good. And each one have their own specific palindromic sequence, okay? So that's that. Obviously, there are mutations that make this even better. So for example, let's say this here would have the same level as usual, and then there are mutations that can even make transcription go even higher because it allows for better binding of the transcription factor itself. So what are these transcription factors really? They are proteins. They have different domains in the protein. So if you remember the tertiary structure, each tertiary structure has kind of a domain linked to it. And so these are all 3D structures. The first one is the DNA binding domain. So the DNA binding domain is where this transcription factor will recognize the DNA and sit on it. The dimerization domain, so these transcription factors, usually they don't work alone. They work with many other proteins. So this dimerization domain allows for binding of 2 transcription factors together. For example, ER alpha, the protein I'm studying, works in homodimers, so it will interact with itself. So it needs this dimerization domain. There's also the ligand binding domain. For example, the estrogen receptor has a ligand binding domain, which will bind, guess what? Estrogen. And so because it binds estrogen, then it can do its job, okay? Same thing with the androgen receptor. It will bind androgen, and it will use that androgen it will be activated by that androgen and then be able to interact with its consensus sequence there, okay? The motif that I showed you right here. And then there's also the activation and repression domain. So this here is where you would have co-repressors and co-activators interacting here. So you have different proteins that will come and bind here, molecules that will bind here, the same transcription factor interacting with itself here, and then the DNA interacting here, okay? So it's quite a complex protein. And then obviously you can imagine in your head that if you remove the ligand binding domain, then you don't need to bind a ligand anymore, and you become constitutively active, right? If you remove the repression domain but leave the activation domain, then you'd end up always active, right? Things like this. If you remove the DNA binding domain, well, you can't bind the you cannot bind the DNA anymore, okay? So that's that. What's important for us is this pathway here. So we'll be looking at a transcription factor called the GAL4 transcription factor, which was found in yeast, and we'll use it as an example for how transcription factors will interact with different genes and activating them and turning them off, okay? For this part here, I'll just show you these videos. So if you go to the online Achieve platform, You can find these videos. They're pretty interesting to watch. They will give you a bigger picture, but I'll try to cover everything in today's lecture, okay? So you can see the different scenarios happening here. Cool? Should be in 12.1. Okay. So now that we broke lactose into galactose and glucose, well, there are pathways to break down glucose, which is known as the glycolysis pathway, but there's also the GAL pathway, which will break galactose into glucose and then use it for glycolysis. And so this pathway here has several genes. There's GAL2, GAL1, 7, 10, and they all have different roles. For example, GAL2 will bring galactose from outside of the cell into inside of the cell. And then you have GAL1, which will convert it into galactose 1-phosphate, etc. We're not in biochemistry. We don't really care about this. What we care about is just the regulation of these genes. And the regulation of most of these genes happens through a protein called GAL4, which is a transcription factor. Okay. So if we first look here at the DNA here, you have 2 chromosomes, as you can see. So that means some of the genes are on the same chromosome, others are not. GAL7, 10, and GAL1 are on the same chromosome, while GAL2 is on another chromosome. And as you can see, the transcription goes in different directions, right? So if you're looking at GAL7 and GAL10, they're using the same strand of DNA to do the transcription, while GAL1 uses the opposite strand to them. Does that make sense? Good. So that's that. So that's the first thing. Now, upstream of these sequences, so for example, transcription going this way, this is upstream of the sequence of GAL7. There are these regions called the UAS regions, the upstream activation site, okay? And this is where our GAL4 will be bound, here, 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 and here. If you can see here, there's 3 of them. Here there's 2, here there's 1, and this is just to kind of tell you that there are different levels of regulation. So for example, here you can have more GAL4 that binds than compared to this one here. And it will also show you which genes are required first and which are not required first. Because if you look at GAL1, you only need 1 GAL4 to activate it. And that's the first one that's kind of necessary. Now, it's not 100% true, okay? But it's just to show you which ones come first and which ones don't, okay? So that's that. There's a more complication to that. And then this GAL4 binds this sequence here. This is the motif that GAL4 will recognize. So technically, if I put this upstream activation site behind a human gene, for example, I use CRISPR or whatever, then I could start activating that gene just by eating galactose, okay? Does that make sense? So you can play with the expression of genes like this by making these chimeras which have, for example, an activation site related to a certain molecule that you have a lot of, for example, galactose. You can start eating galactose. It will be metabolized in your body, but it will activate this GAL4 gene. And then you can put it behind any gene that you want. Cool? Technically, you could put the gene for eye color here and then change your eye color based on the galactose levels, okay? Now I'm seeing some crazy stuff here, but it's possible. We do it in the lab. We use, for example, an activation site for tetracycline, and then just by giving the cells tetracycline, it activates the gene right after. And so we can activate it whenever we want. Yep.
And because like they work in opposite ways, like if that part was activated, it would like allow transcription on both sides.
This one here? I'm not sure of the specifics. If I remember correctly, this one is related to this guy here, and these 3 are related to this guy here. No problem. So I think I said all this here. No. So the GAL4 is, again, as we saw earlier, usually they have a dimerization domain, so that means they come into dimers. So in this case, we have a dimer. This is monomer. Together they make a dimer. You have the activation domain, which I showed you right here. You have the DNA binding domain. So that's this part here. And then you have this dimerization domain here that is not labeled, but it's a dimerization domain. Now this specific transcription factor does not bind any ligand. It's another protein that will bind the ligand, which is galactose, okay? So it does not have a ligand binding domain. So that's that. So this GAL4 is modular in the sense that if you remove one part of it, you will affect transcription. As I told you earlier, if you remove the activation domain, then you will kind of prevent it from doing its job of transcribing a certain gene downstream of it, okay? And here you're seeing the LAGZ gene. This is just a chimeric protein that contains the US with LAGZ, okay? And so because you don't have the activation domain, you don't make the LAGZ, okay? Now, if I were to leave the activation domain but remove the DNA binding domain, same result will happen because you cannot bind the DNA. So scientists like to play with this kind of stuff. So they took a LEX-A site, which is found only in bacteria. Remember, the GAL4 system is in yeast. And so they took a LEX-A site, and they took the binding domain of the LEX-A transcription factor without the activation domain. And then they made a chimeric protein that is half bacteria, half yeast. And they used it to activate the LAGZ transcription. And so you can see this here, okay? This is from yeast, this is from bacteria. And you can do this with pretty much anything. Cool. Actually, some people are, yep. The activation domain is from yeast, and the DNA binding site of the LEX-A is from bacteria. I'll give you just an example of kind of where people are using these. So you can engineer cells to detect a certain molecule, for example, an intoxicant in water. So what you would do is take the ligand binding domain of a protein that can detect that, attach it to a DNA binding site of a promoter that can activate a protein which degrades that, and then you can use it to detect that specific chemical and then degrade it based on this system, okay? If this field sounds interesting, it's called synthetic biology, and people are making like huge pathways based on this, including synthesis of anesthetics and things like this using cells, okay? So you can engineer cells to do this. Take the activation domain from here, ligand binding domain from there, put them together, and play with that, okay? Kind of playing with life, but not really, okay? So that's that. Is it clear up to now? Easy? Okay. So obviously you have to be able to inactivate this GAL4 because you can't always have it on. Sometimes you don't need it. For example, when there's no galactose. Okay. So there's a protein called the GAL80, which will bind to GAL4. So this is a repressor protein, or a co-repressor, whatever you want to call it. And so it will bind to the GAL4 to this site here, the activation and repressor domain. And it will prevent it from doing its job, okay? The only way to remove it is by having galactose in the cell. Galactose, what it will do, it will bind another protein called GAL3, and together they will remove this GAL80 from GAL4 and activate transcription. I know it's getting a little bit messed up because it's becoming a big network, so it's important that you practice this by just drawing it step by step, okay? And if you want to see this, it's good to look at these videos, okay? So that you don't get mixed up. You know what, I'll put this one here so you can just see a little bit. I don't know if there's a sound there. Actually, you know what, I'll let you do this because we need to cover some stuff, but you can just follow these videos and it will show you what comes first, what comes after. And I'll just show you this part here, which is how this GAL80 will repress GAL4. So it will just bind on top of it like this. So to remove it, when you have galactose, it will be detected by GAL3, and GAL3 will come and bind GAL80 and take it off, okay? Does that make sense? So just watch these videos. You'll get a sense of it because it's hard to explain on a 2D slide. Yep.
When it's on the gal, when the gal 8 is on the gal 4,
Yep.
like it requested it because like
Exactly. It will probably prevent it from bringing the RNA polymerase or something like this. I'm not sure exactly the specifics, but it will either it's structural or a functional inhibition. So structural means that it's blocking RNA polymerase from interacting with it. Functional means that it's preventing a certain reaction from happening, and that changes the structure of the protein. Okay? Easy. So earlier I told you, gave you the example of different transcription factors being expressed in different cells. For example, your muscle cells are muscle cells because they have their own set of transcription factors, while liver have their own set of transcription factors. The simplest model that we can use to show this example is the model where we're talking about the mating type of yeast. So yeast have two mating types, either the A mating type or the alpha mating type. And obviously they can form a diploid cell, which is half A, half alpha. That's the only one, just like male and female, kind of. And so if we want to look at the regulation of this, of yeast mating type, we have to look at the transcription factors that are produced in different yeast cells. So in the A type, there's only one transcription factor made, which is the A1 transcription factor, but it's useless. It does not serve any purpose in terms of haploid yeast. Okay, so you'll have this MCM protein, which is another transcription factor that will, a general transcription factor, which will basically bind an enhancer and make the genes that are specific for the A mating type. Now, alpha mating type is a bit different. You still have that MCM1 protein, but you also, the cell also is able to make alpha 1 and alpha 2, and this is where things get interesting. So first, alpha 1 will be bound to the MCM on alpha specific genes, okay? And then the alpha 2 will act as an inhibitor for the MCM protein at the A specific genes. Does that make sense? Or am I losing you guys? You good? Okay. And obviously, because there's nothing bound to the haploid specific genes, so this is genes that are expressed only in haploid state, then they will be on. Now, if we look at the diploid, now you're making basically A1, alpha 1, and alpha 2. And what's going to happen is that alpha 2 will inhibit the A specific genes, same way, because you're not making alpha 1, sorry. Because there's no alpha 1, then you will end up also inhibiting the alpha specific genes. And now because you're making A1 and alpha 2, they will interact together and promote and inhibit the transcription of haploid specific genes. So this is the simplest that you can have, okay? When you're going to do research, it becomes a huge mess and it gets crazy, okay? And so if you have any questions about this one here, it's a good time to ask. Otherwise, yeah. No, just useless. Basically, the mating type locus is different between both cells. In the A cell, it has only one gene, which is the A1 gene, but in alpha cells, it has two genes at that locus, which are the alpha 1 and alpha 2. Okay? But in this case, it's useless unless it becomes a diploid. So it's getting ready for diploid state. Yeah.
So like C would be diploid.
Yeah, exactly. The C is the diploid.
And for A, like the A1, like it can bind to the MC1.
No, it does not interact with the MCM. So it's made for kind of no reason in here. It only becomes relevant when the cell becomes diploid. C'est bon? Yep.
Would it ever make A1 in a healthy cell?
In a healthy cell, no. In a messed up cell, maybe. Okay, so this is gene transcription for you guys. Now, I think we have 3 more slides. So we've seen, again, another level of regulation of gene expression. This is an additional one, and this is where the structure of the DNA comes into play. So DNA, if you spread it, you probably heard this a million times, so we're going to make it 1,000,001. If you take the DNA chromosomes and you open them completely, it's going to be kilometers of length, okay? That's how long it is. So the cell needs some sort of way to make it compact inside the nucleus. And the way it does this is by changing the structure of the DNA molecule by using certain proteins. So the proteins will bind to the DNA, structure it in a way, and that will be called chromatin. So chromatin is your DNA bound to these proteins, okay? And there are 2 different types of chromatin. I will show that in a second. But first, let's look at the proteins that are involved in this process. So they're called histones. There's 4 of them. You have the 5, sorry. You have H1, you have H2, A and B, H3 and H4, and each of these proteins have different regions on them. So these are the protein domains. And these are the types of domains. So you have the histone fold. The histone fold is just what folds the DNA, okay? So this domain is important. The histone fold is important for folding the DNA. Histone fold extension, you can guess that it's extending the fold, right? And then the flexible tails here are pretty useless, but they look like this, right? And so what happens is that these proteins, they will form an octamer, except for H1. So you have H2A, B, H3, and H4. They will form an octamer. So that means you'll have 2 copies of H3, 2 copies of H4, 2 copies of H2A, and 2 copies of H2B. And they will bind to the DNA like this, where you can take a look here. And they will kind of roll the DNA around them, and it will look like this, right? So these are the folds that happen. And then if we look at the diagram here, so first you start with a DNA molecule, then you have your octamer here, and it will enroll the DNA on it, forming these structures called nucleosomes. So nucleosome is an octamer of histones plus that region of DNA, okay? And then these nucleosomes also will roll on themselves and will kind of become like this, and they will make the chromatin fiber. So this way you can turn kilometers of length of DNA into a small little compartment. Cool? So this is a way of regulating transcription and gene expression, because if you have a region of DNA that is kind of folded like this, it's very hard to access with RNA polymerase, and so you will not be able to transcribe that gene. For you to be able to transcribe that gene, you'll have to open up that region. When the chromatin is closed like this, we'll call it heterochromatin. When it's open and accessible by RNA polymerase, we'll call it euchromatin. Okay? I see you all falling asleep. So 2 more slides, okay? And then we're done. So just an example to show you how transcription regulation happens through chromatin structure. You can take a look here at this DNA molecule here, where you have the enhancer, promoter, and the gene. And then forget the insulators, we don't care about them. You'll see them later on. But these nucleosomes, they will form around important regions of DNA. For example, you can see here we have 3 nucleosomes around the enhancer. We have 1 nucleosome behind the promoter and 3 in front of it. Okay, because there's a nucleosome in the middle of the enhancer, you can see that this is going to prevent the transcription factors from binding to the enhancer, and so this will be in a repressed state. Okay. Yep.
Wait, are they all 3 nucleosomes, or just the one that's circled in red?
All 3 of them. But the one that is in red here, it just means that's the one that's on top of the enhancer. Make sense? So this is where technically your transcription factor will bind, but you have something else instead, which is the nucleosome. Now, in the case of promoter here, you can see that there's this NFR region, nucleosome-free region. And because it's free, then RNA polymerase can come and bind here. But then what happens is that you have more nucleosomes on the gene itself, and so the gene will not be really made. Okay. In the case of the active state, you have a nucleosome-free region in the enhancer. So this nucleosome here is opened up, and there's a set of proteins that do that. And so the enhancer is accessible, and because it's accessible, the transcription factor can bind here. The promoter is also accessible, and so the RNA polymerase can bind here, but you will not make the gene because there are nucleosomes that are blocking the gene body. Okay? And so it is active, yes, but you're still not making the gene. There are nucleosomes that are preventing the transcription of the gene itself. Cool? So technically, you would need this region here to open up, kind of, also. And obviously, if you allow for the opening of the enhancer and the promoter, then you remember you get that looping that happens with different transcription factors, including the mediator and TF2D. Okay? Good. So that's pretty much for what's important for you, pretty much it. I just put this slide here to show you the different types of transcription factors that exist. There are a lot more than this. Cortisol receptor is what detects a hormone that is important for stress. So if you have too much cortisol, this is firing nonstop. If you're stressed too much, this is firing nonstop. Androgens, progesterone are involved a lot in cancer, and you can take a look at this. The ones I'm working on are these three here. Okay. Thyroid hormones, if you're doing the test for thyroid, well, this is important for them, etc. Yep. No, I don't even remember that. Yeah, so that's pretty much it. I'll see you Thursday, I guess. If you have any questions, now is the time. Otherwise, for those that have an appointment, we'll go to my office earlier so I can get out. Okay?